Thank you for coming to my poster, Disease Modeling Methods with Cancer Patient-Derived Organoids. I have a few slides to share with you. So this work is based on PDX models established at Matt Burrows' lab at Tulane University. They have 10 different PDX models right now that they've established with different types of tissues from those models. For this work, we're using primary cell culture. These primary tumor cells have been isolated and expanded and then formed into tumoroids. This work, a lot of those done by Margaret Batosian, who's now at University of Chicago. To form this, we used our Puma system. The Puma system is an automated organoid assay system, which uses pneumatic controls to move fluids around in flow chips. It's microprocess controlled, sits in an incubator, and so it's a very convenient system to run assays with tumoroids, organoids. It doesn't do the analysis, but it's compatible with all sorts of downstream analysis, imaging, plate readers, ELISAs, etc. At the heart of this is our microfluidic flow chip. Flow chips are based on a 3D4-well format, so compatible with laboratory automation. But instead of different reservoirs, we have samples which are connected on the bottom by microfluidic channels. And this allows you then to load reagents into connected wells and then transfer them in automatically to your sample. Bottom of it is a clear COC film, so it's compatible with all sorts of high-resolution imaging, and I'll show you some example of that later. For this work, as I mentioned, we're using these patient-derived cell lines. In this case, the cell line was TUBCX4IC, metaplastic breast cancer, the triple-negative breast cancer subtype, and the patient was resistant to Taxol therapy. We took the cell lines, expanded them, and formed them into individual tumoroids using low attachment plates, either single well or multi-cavity array, typically 72 hours to form a nice compact spheroid in there. And then we use a material called nanoshuttle, which allows you to coat them and magnetize spheroids. And this allows you to facilitate manipulation and loading of those tumoroids. One of the nice things about nanoshuttle is it centers the tumoroids in your well. These are 150 to 200 microns. Without the magnetic forces, they would tend to go to the sides of the wells and make it harder to image and analyze them. So for this particular set of experiments, we're doing what we call multifunctional profiling. So you're using different types of analyses to fully characterize the response of tumoroids to different therapies. So we're doing viability assessment, we're doing immunofluorescence staining. In addition, you can do metabolite analysis or cytokine analysis, inflammatory cytokines, et cetera. So the wide variety of assay types that you can do with the Puma system. For the viability staining, we incubated the tumoroids with compounds for 24 hours using the Puma system to transfer fluids in and out, and then add it in the viability stain, and then wash that away to remove background. And here's an example of tumoroids which exposed to two different compounds. You can see the disruption of the tumoroids with increasing dose, as well as increase in number of dead cells, as evidenced by pyridine homodimer. In addition to the viability stain, you can fix the tumoroids and then stain them. In this case, you're using antibodies to E-cadherin and CD44. These are fluorescently labeled primary antibodies. And you can see the very nice images you were able to get. Again, this is a 20x water immersion using Image Express microconfocal system. These are maximum projection images of 2D of a Z stack of images. So really beautiful images showing the localization of different stains to the cells. Similar to viability, looking at response of tumoroids to different compounds. In this case, we're using CD44 as a live cell marker, so you can see the decrease in CD44 positive cells, as well as the disruption of E-cadherin. E-cadherin, very low doses, and so we're looking at this further with lower concentration ranges of these compounds. But for the three compounds shown here, we have very similar responses to viability. And so if you look at the concentration response curves here, percent live versus dead, you can see capturing the EC50 values from that. You can see for the romidepsin trametinib, you saw sort of tens of nanomolar type EC50s. For paclitaxel, it's tens of micromolars, and so a factor of 100 to 1,000 higher than you typically would see for paclitaxel. And this was consistent with the patient's response to Taxol therapy. So a very nice recapitulation of the disease behavior of the patient. So I hope we will show how we can use this Puma system for disease modeling using tumoroids. It also works with different organoids. We can use both organoids formed in low attachment plates as well as those formed in matrigel. So it allows you to do a wide variety of different organoid types up to 1 millimeter in size. And then the multifunctional profiling that enables to characterize on organoid by organoid basis or single tumoroid basis, a wide variety of responses to different therapies. If you have other questions, please don't hesitate to contact me or come to our website. And thank you again for coming to our poster and interest in our work.